design lead. I'm in charge of the user interface and how it interacts with the program.
I'm Emily Wallen. I'm the documentation lead, and I'm responsible for anything that needs to be recorded in written English.
I'm Jimmy Pruitt. I'm the team lead, and I manage the team. I'm Wesley Kozrowski. I'm the testing lead. I just make sure everything's working right.
Our client is Dr. Nirhar, who's a CW professor in the math department, and our advisor is Dr. Hammond. In today's presentation, we will be covering introduction, problem statement, project requirements, lab demonstration, quality assurance, and risk management.
Great, so it's been some time since we last presented. I'll give you guys a quick refresher on what phytoplankton actually are, because PhytoLab is a phytoplankton population modeling tool. Phytoplankton are free-floating photosynthetic microorganisms, and they're present in just about any aquatic ecosystem. They're very important to keep track of because they're a source of food for whales, fish, invertebrates, and all kinds of other marine animals. And the reason that their population levels are important is that when populations grow out of control, it can create toxic conditions for local wildlife. You might have heard of red tide, which is a situation where a certain variety of phytoplankton grows out of control, and the toxins they secrete infect local wildlife, and people that eat them can get sick. Our project was inspired by a modeling tool that already existed, and it was created by our client, Dr. Linhart. It was written in MATLAB originally, and it had a few basic capabilities. First, it could solve differential equations to model phytoplankton population behavior, and it could use those results to build graphs, which could then be saved to the local computer. But it also had a few limitations. First of all, it didn't have a user interface, so anyone who wasn't very familiar with MATLAB and the code involved couldn't really use it. Also, MATLAB is proprietary, which means Dr. Linhart has to keep paying for a license to use it, and that license can be up to thousands of dollars. Also, it had limited forms of input. Users have to manually enter the data that they wanted to use. And finally, it had no web presence, so anyone that wanted to use it had to actually have it loaded on their computer. Which brings us to the problem that Team Phyto is trying to solve. We need to take the modeling tool that Dr. Linhart created and move it into scientific Python. And when we do that, we hope to enhance it in a few ways. We want to create a user interface that's sleek and novice-friendly, and we want to be able to host the application online so that it can be used by anyone easily. So overall, what are the reasons we're doing this? Budgetary, to save Dr. Linhart and the math department money on their MATLAB license. Also for convenience for users, and also to enhance the product and add more features.
PhytoLab has moved a long way since the last time we presented. The last time PhytoLab was able to solve a series of differential equations. It was also able to produce 4 separate graphs, very nice. It was also able to accept several different kinds of input here, but every single one manually if you wanted. It was also able to accept input from Excel files or CSV files. And additionally, we had a clunky but usable interface at that time. Since then, things have moved along and gotten better. We have now increased the number of graphs. We've added 6 more graphs that we can create, so we've got our total up to 10. We have increased the usability, so we've added tooltips to our PhytoLab. So now when people are using it, they can cover the mouse over the different fields, and there'll be a little explanation that pops up, and what is that particular value, and what it does. So it reduces confusion a little bit. And lastly, the interface has been improved to be a little more sleek and user-friendly. So that being said, we're still not done. There's still plenty more work to do. For instance, there's still 2 more graphs that need to be completed. We're in the process of working on those right now. And PhytoLab is looking to be put online. So that's something that we're actively working on. That's kind of going to be our main focus from now until the end of the project in March. So we're actually working with Dr. Linhart and the math department right now to try to get some server space up. So she's helping us out with that.
Okay, so a little bit of background on how the data that we are visualizing is collected. So there's a device called a chemostat, which basically creates a controlled aquatic environment in which you introduce the phytoplankton species that you're working with, be it one or several, and the nutrients that you are trying to measure and the phytoplankton are feeding off of, and a flow rate, since most bodies of water in nature have an inflow and an outflow, so they aren't stagnant. And with this device, you can measure the density of the population of the phytoplankton within it, the concentration of the nutrients or the substrate, and how sensitive the phytoplankton are to that particular nutrient, and the growth rate of the phytoplankton that are feeding off of any particular nutrient. And so before we go into a demonstration, I wanted to show you an example, or what we showed in our last presentation of the user interface. So as you can see, it can only produce 4 types of graphs that were in a list that was not even properly aligned. The checkboxes are all over the place. There's lots of empty white space that could be better served, and there's not a lot of communication as to what these variables are if you're just jumping into this program without previous knowledge. And when you did graph the information, they would come up in separate windows in a mess of visualization. But now, the interface looks a little bit like this. We've implemented, as Wesley said, 6 new types of graphs. They're organized into 3 different tabs, one for population modeling, one for nutrient modeling, and one for something that's referred to as R-star modeling. And population modeling is visualizations concerning the effects of the population of the phytoplankton in response to varying nutrients or varying growth rates. And nutrient modeling is the visualization of the nutrient levels based upon the phytoplankton species and its particular sensitivity to that nutrient or several nutrients. And then R-star, the R-star models are models representing 2 species competing over one or more species competing over one or more nutrients. And it sort of it displays what the balance of nutrients when one would outcompete the other or when they would coexist. And now I will show you the new interface. Right, so as with the previous one, all the inputs go in here. You can use CSV files like so. And now when you mouse over these tooltip or these variable names, it gives you it explains to you what they mean and how they would affect the data, and for each one. And down here in the list of visualizations, you can select from each of these. And for this particular step, we're going to do the R-star plots. And here we have the new image viewer and so instead of it spitting out each image as its own separate window, it now puts them into a single window with these tabs so that you can find the one that you're interested in looking at or go through them one by one, and it's not an organizational mess. And then this is the R-star plot, the final R-star plot here, and it represents this particular one. It shows 2 different species competing over 2 different resources, with the area here in the top right being the balance of resource 1 and resource 2, where these 2 particular species would coexist and not outcompete the other into non-existence. And that is PhytoLab.
On the topic of risk management and quality assurance, continuing from last quarter, we've uncovered a few more risks. They weren't necessarily not present before, but now they've kind of flared up a little more. In particular, unfamiliar language. We weren't familiar. We're now more familiar, but after understanding the math a little bit more, it became obvious that matrix manipulation was more difficult in Python, and that was giving us a problem. Underestimating requirements. We initially thought we could have a database by the end of this quarter where users could save their graphs to and load graphs from, depending on location where they were observing phytoplankton species and things like that. But that is no longer planned. Team members being absent, we just remained in constant communication, and that's kind of a non-issue now, but it's still a risk. And finally, the limited time pre-sub-explanatory. If you're not familiar with the Scrum process, essentially it's a development process for teams of people to work together. And among this process, we've employed stand-in meetings. We're still meeting 3 times a week. That's an extension from last quarter. We thought that was good, so we kept that. We were meeting with our client, Dr. Linhart, every week, hopefully, to continue making improvements. We continue to use a Gantt chart, planning out our whole quarter ahead of time so everybody knows what the task is. Pair programming, everybody has somebody else they're doing the programming with so that problems are solved more easily. And finally, collective code ownership. We all have access to the same repository, and if anybody makes changes, then everybody's responsible for it. Does anybody have any questions?